ETH Zurich researchers have developed a gene switch that can be operated with a green LED light emitted by commercial smartwatches. This revolutionary approach could be used to treat diabetes in the future. Many modern fitness trackers and smartwatches feature integrated LEDs. The green light emitted, whether continuous or pulsed, penetrates the skin and can be used to measure the wearer's heart rate during physical activity or while at rest. These watches have become extremely popular. A team of ETH researchers have now wants to capitalize on that popularity by using the LEDs to control genes and change the behavior of cells through the skin. The ETH team developed a molecular switch that, once implanted, can be activated by the green light of a smartwatch. The switch is linked to a gene network that the researchers introduced into human cells. As is customary, they used HEK293 cells for the prototype. Depending on the configuration of this network, in other words, the genes it contains, it can produce insulin or other substances as soon as the cells are exposed to green light. Turning the light off inactivates the switch and halts the process. As they used the standard smartwatch software, there was no need for the researchers to develop dedicated programs. During their tests, they turned the green light on by starting the running app. The molecular switch is more complicated. A molecule complex was integrated into the membrane of the cells and linked to a connecting phase similar to the coupling of a railway carriage. As soon as green light is emitted, the component that projects into the cell becomes detached and is transported to the cell nucleus where it triggers an insulin producing gene. When the green light is extinguished, the detached piece reconnects with its counterpart embedded in the membrane. The researchers tested their system on both pork rind and live mice by implanting the appropriate cells into them and strapping a smartwatch on like a rucksack. Opening the watch's running program, the researchers turned on the green light to activate the cascade. However, it seems unlikely that this technology will enter clinical practice for at least another 10 years. The cells used in this prototype would have to be replaced by the user's own cells. Moreover, the system has to go through the clinical phases before it can be approved, meaning major regulatory hurdles. To date, only very few cell therapies have been approved.